I believe we finished last time with a conversation about different types of media. We wrapped it up pretty well. I want to just, mention a few words about other less conventional media types, living organisms. So one of them is pretty well known, eggs. Here, this lady is inoculating eggs for the growth of influenza virus. Eggs are used less and less for the propagation of human viruses, specifically influenza virus, for several big, pretty important reasons. First of all, as you can imagine, eggs that are used for the virus propagation has to be so-called SPF, specific pathogen free, right? Meaning that you can't have salmonella in those eggs or any other zoonotic infection associated with poultry. Second problem, they occupy a lot of space. Third problem, yes. Oh, they cannot have. This is what is called specific pathogen free, SPF. So they have no pathogens as far as we know. Okay. They occupy quite a lot of space, which also kind of problem. They're fragile, as you can imagine. That constitutes a third problem. The fourth problem is that where do they come from, the eggs? Where do they come from? Chickens. Can you trust chickens? No, you cannot trust chickens. And it was shown, well, kind of situations several years ago when there was an outbreak of bird flu in the United States. Believe me, it happens quite regularly here, just not publicized that wide. So due to that bird flu, a bunch of chickens and turkeys were dying. It actually got reflected in the prices of turkey it was before Thanksgiving. So turkey prices went up, egg prices went up. But what, in my opinion, was most ironic is that eggs that are used for the preparation of the flu vaccine were in the shortage because of another influenza virus that was killing chickens. So that was a real problem, the lack of necessary amount of eggs. Since then we moved forward to vaccines against influenza that are produced in the cell cultures. For instance, currently the vaccine that is recommended for elderly, but I think you can get it like for any age, is produced in cell cultures. So we become less and less dependent in chickens. For certain filarial parasites, meaning tiny little worms, you may have heard about the disease called elephantiasis. That's the condition associated with the blockage of the lymph drainage, which leads to accumulation of lymph, mostly in the lower limbs and scrotum. So to grow filaria parasites, rodents are used. These parasites do not grow in anything else, since they're worms. Now, some plants even used for the propagation of viruses, obviously plant viruses. That's actually, I wouldn't say it's an issue, but with plant viruses, things become a little bit more complicated. We didn't really talk about the virus infection, right? Like all the processes. I have no intention to do it now. But if you think about it, like the whole idea, virus has to somehow get into the cell, correct? Getting into the mammalian cell, like human cell, isn't big of a deal, because the only barrier that is there is the cytoplasmic membrane. It's not impermeable. in a plant, what you get is you have to cross cell wall. So, and you cannot really cultivate plant cells in a flask. So you only can use the actual plants for the cultivation of plant viruses. You may think about plant viruses as being kind of, not super relevant. Think about viruses that affect citrus trees, like orange trees, in Florida. In Florida, to a little less extent California, those viruses may have same importance as human pathogens. And I'm not even starting viruses that affect growth of grapes in the vineyards. That's totally separate and really, really important story. When I was at University of Nebraska, they had a really good bunch of plant virologists, and there was a joke that plant virologists got more money from the US government than people who studied human viruses that cause human disease, because Nebraska is the huge agricultural state, and for them that was really an important issue. Now, once we inoculated the medium, we place it in an incubator, right? The most important reason why we place it in the incubator and not leaving it here in the bench is that incubator provides us with a controlled environment. That makes sense? Incubator doesn't have to be set on 37 degrees Celsius. For instance, if you want to select your microorganisms for the presence of mutants that are able to grow at the different temperature, you can set your incubator at, for example, I don't know, 42, whatever temperature you want. 42, 25, 27. There are microorganisms that are found in the hot springs or in the Lake Mono in California. Those are pretty hot places. You cannot grow them at 37. They won't. Okay? So for them, the incubator is set for, I don't know, 70, 80 degrees Celsius. The point is, it is controlled environment, which ensures two main outcomes. Outcome number one, your results are reproducible. So for instance, temperature in this room, you can see from here, but it's like something like about 70 Fahrenheit, okay? So you grow your microorganisms here in this room, they grow at one rate. You move them to AP1, which is much hotter, across the hallway, and they're going to grow at a different rate. So results become not reproducible. Incubator increases reproducibility. Another reason to maintain that controlled environment is to compare results between different researchers. If, two scientists work on the same microorganism, and one sees that, his counterpart's results were obtained at 37, he knows what to expect when he grows it at 37. Does that make sense? And last but not least, the practical consideration. Let's say you try to cultivate anaerobic microorganisms. Anaerobic means what they do not tolerate. Oxygen. It would be awfully hard to create an oxygenic environment for them in the room. You put them in the incubator, you can create whatever atmosphere you want. You can increase CO2, decrease oxygen, increase nitrogen, whatever you want. Keep certain humidity. Does that make sense? So that allows you to control the environment. Now, imagine you have those agar plates, and you look at them and you see multiple different types of colonies. Yellow, white, red, large, small. And now you want to isolate a microorganism of interest. here we come to the third step in microbial isolation. We already has got inoculation, right? Followed by incubation. Now we get to isolation. So what happens? What do you do? What are the approaches? So first of all, when we isolate microorganisms, we operate on a certain presumptions. Now, this is really important to understand the whole scientific idea of presumption. Presumption doesn't necessarily truth. Let's say it's going to be cold tomorrow. You presume it's going to be cold. You have pretty good reason to presume that tomorrow is going to be cold, so you're going to be dressed appropriately. But is there a chance that tomorrow is going to be raining and it's going to be 55? Technically, yeah. It's possible, you know. Maybe those guys just, you know, they don't know what they're doing, the weathermen. But your presumption is not necessarily true, but it's most likely correct. Okay? So when we work with microorganisms, we presume that bacterial cell produces a colony, one cell, one colony, which I can tell you up front most likely isn't true. Okay? But for simplicity, we presume that it's true. And second, all cells in the colony are identical, which most likely isn't true either, because when cells multiply, they accumulate mutations, and there is a pretty good chance that cells in the same colony going to have some genetic variability. Does that make sense? But to make our life simpler, and that's normal, reduction, making things simpler, is a normal scientific approach. We say, okay, we think that one cell equals one colony, and we think that all cells in the one colony, all descendants of that original cell, are sufficiently identical. Makes sense, right? Meaning that, hypothetically, if we can pick that one single unique colony, we're going to get a homogeneous population. How do we do it? The first and probably the most important microbiological technique of isolation is called the streak plate. Now, let me explain how it works. So imagine that I'm going to combine between this fancy writing device, the screen, so you will see. Imagine that I put a drop of culture or a sample from a colony onto the surface of the agar plate. And then using a wire loop, usually it's a wire loop, something like that. Okay, here wire loops. Like this. Okay? I streak. I spread the sample over the surface, just part of the surface of the plate. So far, you with me? Then I sterilize the wire loop to make sure that nothing is on it. And I start spreading what I already spread. Does that make sense? So essentially, I am thinning out my original inoculum. Does that make sense? I sterilize wire loop again, and I spread the second streak even more, and then I sterilize wire loop again and spread it even more. before we move any further. Does that procedure make sense to you? Do you understand what I just did? Now what you're going to see. This is your original streak number 1. It's pretty thick. Colonies are all together. Then you grab some of it and you get your streak number 2. Colonies become less frequent, more separated. Okay, and then you start spreading a fragment of the streak number 2, obtaining a streak number 4. Colonies become even more separated. And finally, in the last one here, okay, you get really separated colonies. You may ask, wait a minute, why are we doing that? Like, what's the reason? You already picked a colony. What's the reason? The problem that can occur when you pick a colony is that you look at the colony that is yellow. Okay? Yellow color, single colony. How do you know that it's single species and not a mixture of some orange microorganism and white microorganism growing together in the same place? Does that make sense? You don't really know. By using this technique, the streak plate technique, you can separate two different species and see if your original colony was homogeneous or it had more than one species. Does that make sense? And then, after you performed a streak plate, you can pick exactly what you want. Because at this point, right here, your colonies are separated enough so you know that they are different. And if you're not sure, nothing, prohibits you from doing that again, just to make sure that it is indeed single species. Do I make sense? Now so we're going to do a lab which is going to start next week. It's a 2-week-long lab, lab number 4. Okay, it's one of the big labs. You're going to do the streak plate, and then after you isolate your specific pet, you're going to do more isolation techniques to characterize your microbe even better. Does that make sense? Good. Now, what are other approaches to perform bacterial isolation? It's a pour plate and the spread plate method. Before we get to these methods, we need to understand the concept of the serial dilution. So, how does that work? Imagine that this is your bottle with microbes growing in the medium. Okay, you with me? Okay. Now, each tube contains, now listen closely, just the medium. There's no microbe in there yet. Does that make sense? And each tube contains 9 milliliters of medium. I use 9 milliliters as an example. Okay? So you take 1 milliliter of your original culture and move it to the first tube, to 9 milliliters of medium. What's the total volume? 10. What's the dilution factor?
1 to 10.
1 to 10, or 10 to whatever you want to call it. 1 to 10. Okay? You mix it well. You collect 1 milliliter from the first tube, move it to a second tube. So you dilute your 10 times diluted, 10 more times. Now for the second one, the dilution factor is 1 to 100, or 100x. Does that make sense? You take 1 mil again, move it to the third one, so 1,000 and 10,000 and 100,000 times. Does that make sense? So in your 6th tube, your original sample is diluted 100,000 times. Now you can take that sample, this or this or this, and pour it on the plate, or take a fragment of it and add it to the plate. The point is, the number of cells in each consecutive tube is going to be smaller. So if you will pour your first sample on the plate, you're not going to be able to see any separate colonies. They're all going to be the same. Does that make sense? They're all going to fuse. It's going to be just a massive growth. And as you move down with the dilutions, you start to see separate colonies. And eventually, I mean, I can barely see it here, but there are 2 colonies on this image, okay? Which means in 100,000 dilution, you have 2 colonies. Make sense? Now, this is going to be a part of the next week lab, and we're going to do calculations together. So you're going to work as groups. Each group is going to have your own set of serial dilutions, and you're going to calculate what was the concentration of bacteria in your original sample. Now, speaking of practical application, how can you perform analysis of serial dilutions? Approach number 1 is the pour plate method. This is the one that we're going to do. So what you do here, in these tubes, in all of them, you have liquefied agar. That makes sense? That same stuff that you see on the TSA plates that you inoculate regularly, but this stuff is liquid. It's warm and liquid. So you make dilutions in the agar, and then you simply pour the content of the tube into the Petri dish, swirl it, wait for it to solidify, stick it in the incubator. It's going to grow. Clear? Second approach, which I like more, but for mainly technical reasons, we're not going to do it. It's much simpler. You make all the same dilutions, but this time not in the agar, but in the liquid medium, just the liquid broth. Okay? And then you add a little sample of the medium with, each little sample of each dilution, you add it to the surface of the agar plate. Once you add it, you use one of these hockey sticks to spread the sample on the surface. Much simpler, much less laborious. The main problem is that we have very limited number of micropipettes that we use to add it to the surface. So we're going to do the pour plate method. Any questions about the techniques? You may ask, what do we have to know? We have to match the definition or description to the technique. Am I clear? Any questions? That brings us to inspection and identification. I basically, this is the idea of inspection. So what's inspection? You look at your plate, okay? You look at your plate, and you characterize your colonies. Now, what can you say about the colony? Tell me, please. How can you characterize the colony? Color. What else? Shape in all forms, from the edges to the shape, whether it's, convex, concave, whatever. What else? Huh?
Amount, like how many are?
Size of the colony, very good. You basically covered, there are some aspects to appearance, if it's waxy, if it's glossy, certain significance to it. You can even collect the sample from the colony and stick it under the microscope. And you can do staining, you can do gram staining, okay? For gel or capsular, you know, you can do all these kinds of staining, and you may end up with a characterization of something like large, circular colony with uneven edges, convex shaped, cells are gram-positive bacilli, flagellated, but that doesn't really end the spore forming. It kind of gives you an idea it's probably bacillus, but it doesn't give you a definitive answer. You characterized it very well. Do I make sense? But you still lack the answer. So, what are the methods that are commonly employed to characterize microorganisms and identify? So this is inspection. So now we have 4I. We have inoculation, incubation, isolation, and inspection. Now what's the 5th I? And 5th I is identification. What's the difference between inspection and identification? Hopefully, identification gives you a definitive answer. And I tried, like, believe me, that is still work in progress, but I try to not just give you some obsolete old-timers methods, but give you the ones that are really used. Maybe not, sometimes not as frequent, or maybe sometimes super frequent in clinical diagnostic approaches. Okay? And the first group of methods that I want to talk about is immunological methods. And in order to properly do it, just give me a second, we're going to talk briefly about the immunological basis behind these methods. So first, I want to introduce to you the concept of antigen. Okay? Now, I want you to genuinely understand what we are talking about when we will get to immunological methods. So what is antigen? Can anyone tell me? Go ahead.
It's the substance that an antibody connects to.
Good, but which features does the molecule or substance have to possess to be counted as antigen in relation to
Gotta be foreign, a foreign body.
Foreign, large, I mean reasonably large, complex. So ibuprofen, which is not that large and not super complex, is not an antigen. Okay? The majority of smaller chemicals are not antigens. Proteins, DNA, carbohydrates, large complex molecules are perfect antigens. And importantly, they have to be foreign. Does that make sense? We're good? Now, what happens? What does your immune system do when it, or not only yours, rabbit immune system, mouse immune system, sheep, goat, I just told you the most commonly used animals. Mouse, rabbit, hamster, sheep, goat, horse, 6, okay? So what the immune systems do when they are exposed to something foreign? They will produce antibodies. And antibody is the large protein molecule, looks like a fork, that binds antigen. So right here, antigen is bound to the antibody. Are you with me so far? There are two really important characteristics. There are probably thousands of really important characteristics, but I want to focus your attention on two. First, specific. When I say specific, you can't even imagine how specific. It means that antibody may bind human protein, but it will not bind a monkey protein. Okay, so it's super specific, and binding happens with high affinity. High affinity means that this complex of antigen-antibody is really strong and it's hard to separate. Before we move on, any questions about that whole idea of antigen and antibody? We're good? Now, what does it equip us with? We can identify these complexes. Okay? For instance, we can hang a little detection element that will shine the light, and when the complex is formed and we isolate the complex, we can detect it. Does that make sense? Do you understand the idea of antibody and antigen complex? Now, all the methods that we're going to talk about here, they preceded by developing an antibody. So, which disease you want to use as a model one? I don't really care. Just place a name. Any disease. Anything. Influenza. Perfect. Flu. So let's say we are looking for the flu. Does that make sense? And let's say we are looking for the flu. First, we're looking for the influenza virus in the lungs of a patient that succumbed to some kind of a really severe pneumonia. Okay? We don't know what happened to this patient. We just know that the patient is dead, and we have fresh lung samples. What can we do? First approach that I want to talk about is called Western blot, and I will tell you a bit later why it's called Western. So in this case, we have a piece of lungs, and we grind it up. We add special lysis buffer. So basically, we break down all the lung cells. And as we break them down, we have a bunch of proteins, thousands of proteins. Before we move on, does that make sense to you? Among those proteins are lung proteins and our suspect, the protein of influenza. You with me? Now, they're all grouped together. We cannot really distinguish them. So we need to separate them somehow. The only thing that we know about the influenza protein is its mass. We know how big this protein is. So we take our sample, the lung lysate, and we'll load it on the gel. It is quite literally a gel positioned vertically. You load your sample and you apply electricity. Okay? Now proteins are charged negatively. So where do they go? Where? Down to the plus. Does that make sense? Second, which proteins will move faster? Bigger or smaller? Smaller. I'm going to give you an analogy. I heard it 4th year of college. I still remember it. 1999. Our lecturer said that imagine you have a bunch of kids, fat kids and not so fat kids running through the forest. Fat kids will stuck between the trees. That was his words. Since then, you cannot forget it. Okay? So the bigger proteins will be on top, the smaller on the bottom. Basically, you separate proteins based on their mass. This separation is called, FYI, I'm not asking this, it is called electrophoresis, yes. Now you separated the proteins. Gel is really hard to work with. Imagine that you have to work with Jello. So you need to transfer these separated proteins to some other medium which is more mechanically resilient. So what you do, you line up your gel with a special membrane. So basically you have a gel and a membrane, and you press them against each other, and you apply electricity. Now, look where the plus is, look where the minus is. Negatively charged proteins, where would they go? Tell me, where would they go, negatively charged proteins? Towards the plus, meaning they will encounter membrane on the way. And they will get stick to the membrane. Now what is membrane? What's that fancy word? It's like a piece of paper. It's a delicate piece of paper, but it's not a gel. You can handle it using tweezers, okay, fairly easily. It's not going to like fall apart in your hands. So then what you do, now remember, your proteins are separated. Basically, you're making an imprint of that separation on the membrane. Does that make sense? You place this membrane in the solution of labeled antibodies. These labeled antibodies will produce the light. Well, usually it's fluorescence or chemiluminescence. And once you're done incubating, you put that membrane next to the photographic film. Now here's the deal. The antibodies that we're using are targeted against one specific protein, protein of influenza. Does that make sense? We know exactly where it should be because we know its mass. Correct? So if we know its mass, say we know this is where it's supposed to be. So we'll look at our samples and say, aha, we have influenza only in the first line. In two other lines, in two other, no influenza here. Yes, flu. Okay? Does that make sense? We managed to detect it because we know exactly where this protein is supposed to be. Now, is this method used in clinical practice? Yes, but not frequently. Main problem with this method, it is really hard to quantify. I mean, you can compare two samples. You can say this sample has more than another, but it's hard. That's problem number one. Does that make sense? Problem number two, it takes a long time. So in the best times when I had like samples of tissue lined up in front of me, and if I was focusing only on Western blot, I could start at 7 a.m. and finish at 7 p.m. One analysis, about 12 samples. Okay, it takes a lot of time to prepare, a lot of bench work. This is why I love Western blot, because it's a lot of actual hands-on experience. It's a really cool method. And also, when you have gel running, transfer happening, incubation happening, it's free time. Like you're sitting next to it doing nothing. If somebody will tell you, why are you doing nothing? You tell them, no, I'm working. You can have lunch, you can have coffee, but I'm working.
Or supervising.
Huh?
Supervising.
Sort of, yep. Actually, nobody will let you sit next to the Western blot again and do something else during that time. It's actually pretty low maintenance approach. You know, now companies even produce gels. You just take them out of the package and they're ready to go. Because usually people make them themselves, but now you can just buy them. The advantage, the biggest advantage, it's uniquely sensitive. We're talking about 10 to the power negative 12 grams per sample for a protein. It's insanely sensitive. Okay. Well, here's another disadvantage, but that's a disadvantage for many methods. Think about this. If you do not find influenza protein in your lung sample, does it make you closer to the answer, what was the cause of death? Well, not really. You just can say, okay, this guy didn't have influenza, but we still have no idea what he died of, right? So that's the problem. I always say you look, you search, no. You can find only what you are looking for. You cannot accidentally stumble upon something else. Okay? Does that make sense? Now, a little bit different technical solution is used in ELISA. So in ELISA, and I want to focus your attention on this sandwich ELISA on the right here. In ELISA, and ELISA is the acronym, it's not a name here. You have an antibody that has been pre-adsorbed on the plate in the wells. So when you add your sample, lung lysate, if your lung sample contains the influenza protein, that protein here, the antigen, will bind to the antibody. Then you can add another set of antibodies here that will manage to detect your antigen. Does that make sense? Basically, we call it a capture ELISA because this antibody captures the antigen. And then you can figure out whether the antigen is there. Now, what's convenient about ELISA compared to Western blot? First of all, it can be quantified. Think about this. The more antigen you have bound, the stronger the signal you're going to have. Is that clear? More antigen means more complexes, more signal. Signal can be color change or fluorescence or luminescence. So it is easily quantifiable. Okay. Second advantage, it has high processivity. You can do ELISA manually. When you do it manually, it's 96 well plate. Plate is about a little bigger than my palm. You have 96 wells on it. Put, say, 16 wells on the standard and controls, stuff like that. You have 80 wells to run samples. Counting two, in repetition, you do each sample in repetition. You can run up to 40 samples on one plate. That's a lot of samples. Moreover, some maniacs do not 96 well, but 384 well plate ELISA. Which, insane. I don't know how people do that. No, seriously. Because wells are really small and the size of the plate is the same. Now, 360, 380, 760, 1520. So there is a plate that is 1536 wells. That's not for manual. That's for machines. This is another advantage of ELISA. It is very easy to make automatic. To the point when you have all your samples lined up in the tray. That makes sense? You load the tray in the machine. The machine already has all the antibodies. It has a plate. It has all the buffers used to wash the plates. Everything is there. So you load the tray and you push the button. The machine will load the samples, will incubate them, will shake them, will wash them, everything will be done. These machines are not cheap, but they're super convenient. And they're used in big clinics when you have like thousands of samples coming through. That makes sense. But again, the problem with the LISA is that you get the signal only for what you're looking for, right? You cannot detect something else. Now, let's say our LISA with our lung sample was successful. So we know that this gentleman died of influenza in the lungs. It would be lovely to see where was the infection. Was it on the apical side of epithelium or basal side of epithelium? Was it affecting type 1 alveolar or type 2 alveolar? Was it in macrophages? Was it in endothelial cells? Where was it? For this we can use immunofluorescence. Okay? So imagine you have a cell. And the cell is infected. Depending on the type of infection, it may carry antigens outside or sometimes inside of the cell. Does that make sense so far? If it's inside, you can treat the cell and it's called permeabilization. You can make membrane permeable. But the point is, you add your antibodies and they detect, okay, they detect your antigen on the surface of the cell or inside the cell. Now, what is the advantage? Well, look at this picture. It's a friend of mine, really good friend of mine, sent me this picture. It's an example of immunofluorescence. You know, red cells are neurons, green cells are astrocytes, and the nuclei are colored blue. You can make different colors, use them in the same sample. So for instance, if we go back to a patient with a lung, possible lung infection, you can stain for influenza virus. For macrophages, you can find specific marker for macrophages. And for type 1 alveolar cells. And you can see where the virus is. Does that make sense? So that this method isn't quantitative really, but it allows you to identify where the infection is. Does that make sense? And of course, antibodies here, they should be fluorescently labeled. So they have a special, actually, there is a guy back in Moscow that is a really good chemist. And he's making different fluorescent labels. He has hundreds of manuscripts published because he makes a new fluorescently labeled antibody, and then people say from United States, he has collaborations all over the world. They call him and say, can we have this antibody? He says, sure, I'm on the paper. So they publish a manuscript, he's on the manuscript. Publish another, he's on the manuscript. The guy's, he's a great chemist, but it's a nice way to get publication. Now, for a long time, I was kind of struggling with myself. This page perhaps looks absolutely overwhelming, but it is one of the most state-of-the-art methods that is gaining a lot of steam in clinical diagnostics of bacterial infections. And it is called, don't get scared, MALDI-TOF /MS. Now we're gonna deconstruct the name. The name means matrix-assisted laser desorption ionization, time of flight, mass spectrometry. It's a lot of fancy words. And I will do my best to break it down and make it really, really clear. So first of all, what does that mean? Matrix assisted. Here, you see that yellow stuff with little dots? This is matrix. So imagine you have your culture, bacterial culture. Am I clear? You have your bacterial culture. You mix it with matrix and you apply it on the plate. That is it. Okay? The role of matrix is to basically bound your sample and make it stick into the plate. That's it. Next step, laser desorption ionization. Let's break it down. Well, you all know what laser is, right? Really powerful beam of light. No, seriously, that's the whole. Really powerful beam of light. Carries a lot of energy. Okay. Now, desorption. Imagine that you have a pile of snow. You throw something and snow just flies up. Basically, you desorb the snow. Okay. Ionization. Some chemical particle usually loses electrons and becomes charged. Let's see how all these three things come together. Here's your matrix with your sample in it. Yes? Desorption. Well, think about, Lisa, think about you have a pile of dust. You throw something, dust flies up. You basically desorb the dust. So in this context, desorption is to kick something out so it flies out. Does that make sense? Sure? We good? So,
I'm still lost on that. Desorption.
You have some really densely packed chemical. You perform certain impact on it. The chemical is relieved from that package, just flies out, just gets free. Okay, that's desorption. It's a fancy word for letting it loose. Make sense? So look here. So this green lightning is laser. It hits the matrix. A lot of power, right? So all the stuff in the matrix, including your sample, gets in some kind of vapor. It's not really vapor, but close enough. Does that make sense? It flies around. We good? Is that understood? Don't look for something more complicated than I'm trying to tell you. Okay? So it all flies around. Not only it flies around, the energy of the laser made your samples, fragments of your sample, ionized. So they now have a charge. Do I make sense?
And that's what the beam, the laser did?
Laser beam, exactly, very good. Laser beam hit matrix so hard that fragments of your sample that are in the matrix, they start to fly out, and as they get hit, they also lose electrons. So they become ionized. So laser, look at the title, laser kicks them out, desorption, and ionizes them. Does that make sense? Now you have a bunch of ions that have A, different mass, B, different charge. Right? You have all kinds of ions. And they fly into the accelerator, which is basically a giant electrical magnet. Okay? And all these ions that have different mass start to gain speed. You see them flying. Now look at the picture. I know it's rather small, but which ones are flying faster? Small ones. But remember, they differ not only in mass. What other parameters also differ in between those ions? Charge. So, the time it will take them to fly from accelerator to the detector, and they get reflected at some point. So time that it takes them to fly, so-called time of flight depends on which two parameters? Mass and charge. To tie them together, we put on the x-axis mass to charge ratio. So we take a mass and divide it by charge. Does that make sense? So if you have a fragment which mass is 8,000 and the charge is 1, it's going to fall here. If you have a fragment which mass is 8,000 but the charge is 2, it's going to fall in here because it's going to fly much faster because charge contributes to it. Does that make sense? So basically, here's the thing. I'm going to kind of sum up what's going on before we get to all these fancy graphs. Okay? What you do, you take your sample, you blast it with a laser. Laser pushes the fragments of the sample out, turning them into ions. Are you with me so far? These ions get accelerated and they fly towards the detector. And time of flight will be dependent on the ions' mass to charge ratio. And different ions with different mass to charge ratio will give you these different peaks. Does that make sense? They will give you different peaks when detector, detects them, basically. Okay? Now, it all sounds pretty cool, but the question remains, how the heck can we use it for identification of microorganisms? Imagine that you have a pure culture of Bacillus anthracis. Basically, it's a bunch of proteins. Okay? You mix this pure culture with the matrix, do this whole shenanigan, and you get a very specific set of peaks. Do you see this very specific set of peaks? And it means that every Bacillus anthracis is going to have very, very similar set of peaks because they all have very, very similar set of proteins. Do I make sense? Basically, you produce a molecular signature. And first that I want to show you, here you have 4 different species, 2 gram positives and 2 gram negatives. They all are remarkably different. Even these 2 species of bacilli have very different set of peaks that do not overlap a lot. Does that make sense? However, if you will take, this is the control, this is Proteus mirabilis. We know that on the left is Proteus mirabilis, so you know these are certain peaks. And on the right is an unknown sample, which looks very much alike, and when it was independently tested, it turned out to be indeed Proteus mirabilis. So, what do we do now? How is it used in the clinic? You have a culture of a pathogen that you suspect caused the disease of a patient. You load this unknown, absolutely unknown sample here into the matrix and you run the machine. In 2 hours, your machine will not only produce a graph like that, it will compare the graph to the database of the known bacterial species and will tell you what is most likely you're looking at. Does that make sense? So the whole analysis from loading the culture to getting the answer takes about 2 hours. This is awesome. First of all, because you do not find only what you're looking for. You genuinely have no idea what this culture is. And if the matching microbe is in the database, you got it.
Kind of like a fingerprint.
Absolutely. Absolutely. It's a fingerprint of bacteria. That's a good comparison. Make sense? Now, obviously, if it's not in the database, then it's not going to work. The limitation is that you need to have a pure culture, although I've heard some reports that they started to work on using patient's blood. So they would be able to distinguish, to kind of isolate the characteristic peaks using unpurified sample. I don't know how it's going to work, but so far you need pure culture. Second limitation is the price of the machine. It costs 750 grand. So only biggest hospitals use it because they need to have enough samples to justify the purchase. So, for instance, like I'm not sure, I'm not positive, but I believe it's likely that clinic or UH, I don't know who handles, I believe clinic handles more patient. Clinic probably has something like that. Okay, because they cover a huge area. Does that make sense? Now, couple last methods that one I want to briefly describe what the idea is. So, genetic methods. First one is PCR. This is by far is most widely used method of identification of anything from viruses to protozoa. All organisms. With viruses, it's slightly different, but let's say you have organism with the DNA genome, okay? Double-stranded DNA genome. Are you with me? Now, these genomes are quite unique, meaning that, let's say, our genomes, are they very similar? Yeah, they are. Okay, they're very similar. Our genomes and genomes of chimpanzee, are they different? Yeah, somewhat. Yeah, somewhat different. Yeah. And we can actually find this difference. We can say, okay, this particular part of the gene, this sequence, we'll call it a sequence, the sequence of the gene in humans and in chimps is different. We can say the same about different, say, bacteria. We can say that E. coli has its own unique DNA sequences that distinguish it from, I don't know, Yersinia pestis, from Enterobacter genus, from Salmonella typhi, and so on and so forth. Am I clear? So they all have these unique fragments. And the goal of PCR is to detect these unique fragments. How does that work? First, we need to identify, we need to know what is the sequence. So say we know the entire genome sequence of E. coli. And we say, all right, here's the deal. We know that this part and this part of the DNA are unique to E. coli. So we're going to produce complementary short molecules, short DNA fragments, shown here in red, called primers. These primers will bind only to these unique sites in the DNA. Are you with me so far? Only there. They will not bind to DNA of Proteus, influenza virus, Giardia lamblia, or human. No, they're unique for E. coli. Clear? And then we're going to heat up the DNA of E. coli, or our sample that we suspect is E. coli, and we're going to add primers, and primers will bind to this individual DNA strands. And then we're going to put an enzyme there called DNA polymerase that will start extending each primer, completing a double-stranded DNA molecule. Okay, so basically what we're going to do, we're going to synthesize a DNA fragment that corresponds to the space between the primers. So you have two primers, look at me, you have two primers, and they start to extend. And now you have short DNA molecule that is a copy of the genome fragment between the primers. Does that make sense? So basically, like in the book, you say, okay, you're going to copy from here to here, and you're going to copy it 40 times. Does that make sense? Now, it's not just 40 times. Look at this. How many DNA molecules you have here in the beginning? How many DNA molecules, double-stranded DNA molecules in the beginning? How many molecules here, double-stranded? One. How many double-stranded DNA molecules you have here? Two. That's one round. After second round, you have, after third, 8. After 20, you're going to have about 1 million original, gene. After 45, it's like trillion and a change, okay? Like 500 trillion copies. So basically, you can amplify one fragment of the DNA trillion times and then detect it. You can't detect just one little copy, but you can detect trillion little copies. Okay? So this is an extremely powerful, extremely specific, extremely sensitive tool. By now, we can detect one molecule of DNA in a sample. Like literally. You take sample for analysis, 20 microliters, you can have one DNA molecule there, and it will be found. It's unbelievably sensitive. Again, the problem, you find only what you're looking for. Makes sense, right? Because if you take different primers, it's not there. Sorry. Now, so PCR is like to-go thing in clinical practice. Anything is tested by PCR. Okay, you go for like HIV test, it's probably going to be PCR, the first one. STDs, PCR. Okay, good clinical influenza test, PCR. It's all going to be PCR. Okay, like to clinically confirm the first results of the swab, PCR. Can you get around the idea of finding only what you're looking for? Yes, you can do it using sequencing. Now, sequencing for me is magic. I tried to figure out how it works. I said no. I mean, you can get through it, but I realized even if I will figure it out, I won't be able to explain it to anyone. It's really complicated, technically and chemically. Basically what happens, you isolate a bunch of DNA and you stick it in the machine, and after a couple of days, sometimes less than that, machine gives you sequences of all DNA fragments that you have. Everything, every single gene, everything that you have there. Okay? So you're not finding what you're looking for, you're finding everything, which is one of the major hurdles. Because if you have a patient who is sick, okay, and you collect, I don't know, throat swab, patient has like recurrent respiratory infections, you collect throat swab, you stick it into the machine, you get millions of genes, and now you have to sit down and figure out what it is. I mean, you can subtract human genes, you can subtract common bacterial genes, but you probably will still end up with 20 to 30 suspects. And you have no idea which one to blame. So, sequencing can be a little bit overwhelming, but main advantage, it kind of narrows down the field of search, okay? And it gets cheaper and cheaper. I'm waiting. Illumina, a year and a half ago, Illumina promised to put the machine on the market that will sequence human genome for $100. When it happens, I guarantee all offices, physicians' offices in the United States will be equipped with these machines, and all newborn babies will get their genome sequenced. Because that would be just absolutely terrific. You know about all potential genetic abnormalities from the get-go, and you can address it timely. And you can also do things like, now say the problem, say I do have chronic rhinitis. I have no idea why it is. Okay. If that would be the case, I would go to ENT, they would swab my nose, they would stick it in a machine, they would tell me, okay, you have this set of microorganisms, these 5 look really suspicious, and among the good ones that we know about, I don't know, you don't have these 2 good ones. So you have like screwed up microbiome in your nose. This is why probably, you have chronic rhinitis. Let's think maybe we will transfer some of those species. We'll try to repopulate your nasal microbiome. That opens a lot of doors for us. That makes sense? So we're going to wrap it up.